Biotechnology is the term used to describe a field in which scientists make controlled use of living cells to perform specific tasks. Genetic engineering is the term used to describe a subset of biotechnology that's involved in the manipulation of DNA for human purposes. Some uses for this is to produce drugs and hormones, such as growth hormones that may be lacking in an individual, to improve treatments for diseases for humans, as well as to increase food production from plants and or animals for human consumption. Recombinant DNA is the term used to describe DNA that's made from two or more sources. Recombinant DNA is created when a gene of interest, one that produces a useful protein or trait, is put into another piece of DNA. Steps in genetic engineering: the recombinant create the recombinant DNA. Restriction enzymes are used to cut the gene of interest, the one that we want to use to make a specific protein, out of the original DNA and splice it into a vector DNA. Plasmids, bacterial plasmids, are common vectors. Plasmids are circular pieces of DNA that exist separately from the bacterial chromosome. Plasmids are small circular pieces of self-replicating DNA that exist outside the bacterial chromosome. So in the top image, you see the vector DNA and the box around the sequence recognized by a specific restriction enzyme. This restriction enzyme is going to cut the DNA at this specific sequence. These ends, these sticky ends, just like DNA replication when it's separated inside the nucleus of the cell where nucleotides are going to bind, these sticky ends will allow the binding of complementary nitrogenous bases. The DNA fragment from the donor cell and the source of the source DNA with the gene of interest. You see the sticky ends on both. Fragments form temporary bonds through base pairing, A and T, C and G, and you end up with the recombinant DNA, where another enzyme, the DNA ligase, permanently bonds these fragments. Vector, which is these recombined plasmids, are taken up by a bacterial new host cell. The bacteria containing the gene of interest, the protein that we want, is identified and isolated and produce numerous copies of the gene, amplifying this gene. Bacterial cloning or through the polymerase chain reaction, abbreviated with PCR. In PCR, the DNA of interest is unzipped by breaking the hydrogen bonds to form the single strands. These single strands are mixed with primers, which are special short pieces of nucleic acids that pair with the bases complementary to each strand. So these primers serve as the start tags for DNA replication. Nucleotides and a special heat resistance DNA polymerase is added to the mixture and then cooled to allow for the base pairing. The procedure is repeated numerous times, and each time the number of copies of DNA is doubled. And this process is used such as in criminal investigation. So this image shows where step one, the DNA of interest, isolate the DNA from the two sources. The plasmid is that circular piece of DNA, bacterial DNA, that exists outside the bacterial chromosome. And then we have the cell with the gene of interest, the one that we're wanting to amplify. In step 2, both DNAs are cut with the restriction enzyme. When mixed, the DNAs are going to recombine by base pairing. DNA ligase enzyme links the new gene into the plasmid. This recombinant plasmid containing this gene of interest is placed back into the bacterial cell, and the bacterial clones contain many copies of the donor genes. And so at that point, we identify the recombinants containing the desired gene, clone and harvest the gene and or its protein products. This is the image of the polymerase chain reaction described earlier. Genetic engineering is a term used to describe a means to produce large quantities of desired proteins. For example, growth hormone or insulin. It allows a gene for a desirable trait to be taken from one species and transferred to the cells of another species, making a transgenic organism. Salmon are used to produce growth hormone. Bacteria are used to produce insulin. Insulin is a hormone secreted by the pancreas that causes cells of the liver, muscle, and fat tissue to take up glucose and store as glycogen for energy reserve. Genetic engineering has been used in corn to deliver radioisotopes killing cancer cells. Genetic engineering has been used in bananas to make an edible vaccine for hepatitis B. Genetic engineering has been used in soybeans to make a vaccine for the herpes simplex. Applications of genetic engineering may be environmental, such as oil-eating microbes that can withstand ocean conditions, which would be something that might have been used in Hurricane Katrina. It's been used in livestock for cows to produce more milk. In pharmaceuticals, known as gene farming, notice the pH, where transgenic animals produce a protein with medicinal value in their blood, milk, or their eggs. They're then collected in the purified protein. This has been used, this gene farming has been used for cystic fibrosis. Cystic fibrosis is a genetic disease that causes thick, sticky mucus to form in the lungs, leading to respiratory problems or death. So this has been tested using this gene farming as a drug to help prevent cystic fibrosis. In sewage plants, genetically engineered microbes remove excess phosphate and nitrate discharged into waterways. Excess phosphate and nitrate can result in excessive aquatic growth, which could choke waterways or dams. It could cause excessive algae growth, which could produce chemicals that may be poisonous to fish and livestock. Proteins have been engineered into sheep to treat people, as previously noted, with cystic fibrosis against this lung damage. Applications of genetic engineering. The raw materials, goats and bacteria genetically engineered to produce spider silk proteins. Spider milk is 5 times stronger than steel, yet still lightweight. Applications for this could be in bulletproof clothing, thinner surgical thread, stronger yet lighter weight racing cars and aircrafts. In agriculture, crops have been genetically engineered to be resistant to pests, to be more nutritious. For example, golden rice produce high levels of beta-carotene, a precursor for vitamin A, which could be useful in countries where more than 1 million children worldwide suffer from vitamin A deficiency, and 500,000 go blind each year. In this image, we see where a gene of interest is put into. In this example, it shows the sheep. Implant the cell into the host mother. Identify the transgenic offspring with the gene of interest. Gene of interest is expressed maybe in the milk. The milk's collected. Milk proteins are separated, and the useful protein is isolated. Gene therapy puts functional genes into body cells affected by a defective or mutant gene. And viruses are used to typically deliver the healthy gene, because viruses typically attack only one type of cell. Gene therapy has the potential to cure inherited diseases. There's been some success with severe combined immunodeficiency disease, SCID, and X-linked SID, which is a mutant gene on the X chromosome. More than 4,000 human diseases have been traced to defects in single genes. There's hundreds of clinical trials that exist, although to date the FDA has not approved a gene therapy. This severe combined immunodeficiency disease was the first condition to experimentally be treated with gene therapy. The mutant gene prevents production of an enzyme necessary for white blood cell maturation, thus a compromised immune system. So this shows some promise that gene therapy can hold, where we can possibly cure some human diseases. Genome is the term used to describe an entire set of genes carried by one member of a species, whereas genomics is the study of the entire genomes. So the goal of genomics is to determine the location and sequence of genes. Many have been created to automatically sequence the DNA. Supercomputers have been created to automatically sequence this DNA. The Human Genome Project was completed in 2003. It resulted in the knowledge of the location of genes along all 23 pairs of chromosomes. Current knowledge on specific location exists for more than 1,400 diseases, such as Duchenne muscular dystrophy, cystic fibrosis. So this allows the gene testing for family members. The human genome consists of 20 to 25,000 genes, and we're 99.9% alike in our DNA sequencing, and this 1% is what we want, this 0.1% is what we want to gain a greater understanding of, because this 0.1% or this 0.1% that differs from person to person can lead to the understanding why some people develop heart disease, Alzheimer's, etc., and others do not. Microarray is a tool that's used to analyze gene activity under different conditions. In microarray, they take thousands of DNA sequences and stamp it onto a single glass slide, a DNA chip, So it can be used to identify genes, say, that are active in cancerous cells but not in healthy cells. So this is an image of a microarray. So if scientists can use this to see what genes are active in a cancerous cells versus the genes that are active in a healthy cell, then this can lead to genetic engineering, where we can possibly modify, identify these genes, and find a treatment and a cure. Comparisons of genomes of different species provide evidence of common evolutionary paths, in that we share 50% of our genes with fruit flies and 90% with mice.